Hey Biology, this is Mr. Hausman, and one of your assignments for this week, as we continue the study of cell division, is to do a little review and recycling of knowledge about this picture. Now, this picture has a lot of details starting in the upper kind of top left corner, maybe around 11 o'clock. You notice that there's cells that have chromosomes or DNA that looks like spaghetti. And I'm referring to this region here. And that's how a cell looks most of the time, and that's interphase. Well, as things get ready to divide later on in the cell cycle, the chromosomes become visible, and that is probably prophase. This is a little bit after prophase and before metaphase. Some people call this prometaphase. You are not responsible for that, but hopefully we remember metaphase, middle. Then we have anaphase, telophase, and finally cytokinesis. And it leads to making duplicate copies of cells. And that's what we're going to review today. So you're hopefully already completed the questions on page 10 and 11 in your first mitosis and cell cycle packet, the first packet on cell division. So it's up to you if you choose to watch the pretty cool video on mitosis. The Amoeba Sisters have a great one, as well as several other YouTube channels. The link that I might have sent you to actually might be broken. Sometimes videos are no longer available. We're not going to watch that in this clip, though, but the URL at the top of this, if you want to write it down, QVC JD, capital N, lowercase x, capital J, lowercase re, capital E. That's actually how YouTube identifies the clip, and that's how you could find it pretty quickly. Or you could just Google or YouTube search cell cycle and cancer and Amoeba Sisters, right? That's a channel we know. So the very first part of this review is to do a quick kind of naming review, vocab driven. And DNA is a molecule you know well. It's made of sugars, phosphates, and nitrogen bases. But how the DNA is organized matters a great deal. So A is kind of like the most fundamental level, right? So A is a picture of, right, loose DNA. And we would refer to that as chromatin. It's not organized. It's just kind of sprawled out like spaghetti, kind of like anywhere in the nucleus of a cell. But as the need presents itself, as a cell gets ready to do cell division, the DNA automatically starts winding around these cores. And it looks like it's almost like a spool of DNA, and that's what it is. But it only wraps around each of these 4 times. And those are proteins called histones. Histone proteins. And that's how the DNA starts to condense. It gets more and more tightly coiled. The histones, you can actually see them kind of like bunched together. And then we get to step C. Well, step C, or picture C, is now DNA that's highly organized. We call it condensed, and that represents a chromosome. Finally, what's going on at D? Why are we pointing to the 2 arms or the 2, some people call them legs of the DNA or the chromosome molecule? And those are referred to as sister chromatids. Well, they're really identical copies or twins, and replicated DNA is the result, right? So when you have replicated DNA, you have chromatids. Okay, so quick recap. Chromatin is loose, chromosomes are organized or condensed, and then finally chromatids are the 2 copies of replicated DNA. They all begin with chrome, C-H-R-O-M, but they each sound differently. Chromatin, loose, chromosomes, X-shaped, condensed, and then chromatids are the 2 arms. Okay, now let's go to a slightly different body of knowledge. Let's go over here to the cell cycle itself. So we started with DNA as a molecule. So interphase takes up this entire region. Let's go over to our pen and let's color it in black. We'll change to black. And interphase, right, is this whole region that begins right here at A. Interphase keeps going through what we're going to identify later as phase B and all the way around to C. I think we've seen this numerous times. We've discussed it. It's been visualized in class. And when you talk about the life cycle or the cell cycle, right, of a living cell in your body, most of the time it's in interphase. So we'll call that, black color, right, the black line on the outside. But within interphase, you should know that there is G1, and I want you to make sure you have both its symbol, G, and it's called growth phase 1. B, which is a little bit exaggerated. B, the second part of interphase, is the S phase, and that stands for synthesis. What are we doing there? We're making DNA copies. We're making the sister chromatids. Make the copies of every chromosome. And then C is called G2. It's the second growth phase, but I refer to it as the getting ready step. That's its nickname. That's how you can know what you're talking about. You're getting ready for what's going to happen next, which is, of course, these part D. So let's say we've got A, B, and C. Where's the DNA replication? Right, that's at B. Let's write that in. Right, and then what are the growth phases? That's A and C. So you hopefully have noticed now G1 and G2 are not next to each other. They are slightly different in their appearance, but this is the cell going through its normal everyday events. But B is the standout event. This is the DNA replication, and we refer to it as S phase. Okay, so quick review there. Let's keep going. See what else you remember, hopefully strongly. Let's go to the next slide, if I can remember how. Now I want you to focus on just D. And D, I want to be very careful of. A lot of people just say, oh, that's the mitosis phase. Yes and no. D is referred to as cell division. And it's made up of, right, this whole thing, cell division, is made up of different subparts. So we've already labeled A, G1, B is S, and C is G2. But now let's get to E. Well, suddenly you get into the cell division step, right? This is all the red part of the cell cycle, from the end of G2 until the cell goes back into G1. And E is referred to as prophase. Prophase is when the DNA prepares, right? Pro and prepare. F, what comes after prophase? I remember that it sounds like something that Jothniel taught us about PMAT. So I know that once I get into mitosis, picture maybe a dog, having an accident and peeing on the mat, like a doormat or they're called pee pads for little puppies. So there we have prophase, then we have metaphase, then we have anaphase, and then followed by telophase. Maybe I've been pronouncing it most of my life. I still sometimes call it telophase, but telophase. And you think you're done because those are the 4 letters of mitosis, right? Mitosis is made of prophase, metaphase, anaphase, and telophase. And that's true. But then there's cytokinesis. Technically, it is not part of mitosis, but it is the final step where the cytoplasm, you can tell what's going on there because cyto is in the beginning of it. Kinesis means move. So where cytoplasm is splitting, it is moving, the cell membrane pinches off. So those are the answers to that page. Let's keep going. Okay, now, based on our laboratory experience, based on our knowledge of these phases, these are, of course, cartoon models. And I want you to do kind of what we did in lab, where you identify cells in each phase. Well, the easiest thing to do is to find maybe a cell in one phase and then do comparisons. So I'm going to call attention to the top row. Let's take a look at the top row. So there's cells doing all different things. Sometimes the chromosomes are lining up. Sometimes they're visible but not organized. Sometimes they're actually kind of in the middle of or ending their division, right? You can see a new cell wall forming. But then let's take a look at cell 4. Well, 4 is exactly what you think it is. 4 is a cell just living its cellular life, making proteins, just doing its thing. And that's interphase. Well, interphase, right, is also going to be 5 and 6 because they look the same, and 8. And there's many of them. In fact, usually it's over 90% of the cells. So I'm going to keep going and write them all in. And 19, and then 20, and then 22. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 out of the 22 cells are in interphase. Prophase is a much shorter step. And where can I see chromosomes that are organized, visible, but not lined up or pulling apart? And that draws my attention to right here. A cell that has no chromosomes lined up in the middle, but they're also not clearly in the process of pulling apart. They're just kind of in a nucleus, and that happens when the cell is getting ready. That's prophase. It's preparing. I think 15 looks the same. I think 21 looks the same as 2 and 15. So we have 3 cells in prophase. Where's metaphase? Well, I remember that there's a mnemonic device that M stands for middle. The chromosomes line up in the middle of the cell. The spindle fibers line them up and attach, and I see, and even though there's no spindle fibers shown, clearly cell number 1 is metaphase. Cell number 10 is looking the same, and then way out at 17 in the 4th row. Anaphase is just numbers 7 and 18. That's the only 2 I see. And notice that anaphase is very quick. There's only 2 cells that are in anaphase. Telophase, in a plant cell, you can tell because it looks like the beginning of a new cell wall. And as I just circled, only, let's do yellow, although it won't show up great, 3 and 9. And it says to add some spindle fibers, so I'll do that. I'm going to make them black. Spindle fibers for these cells, right, would connect to the poles or the ends of the cell. Now, in anaphase, like I'm drawing right now pretty quickly, notice there's no spindle fibers in the middle because they are shortening. They are basically contracting like a muscle fiber, and so we'd have spindle fibers here as well. I think I drew all of them up, missed 1 and 17. They look like whiskers, okay? And I think that's all the cells that should have visible spindle fibers. Let's move on. Here it is. Phases of cell division. Can you put them in order? So who's number 1? Number 1 is probably cell Y, right? Cell Y, that's a cell that has no visible chromosomes, so that's interphase. We usually start with interphase. And so I guess that's not cell Y, it's cell D. Y is pointing to a structure. Then you can see the chromosomes become visible. Where do the chromosomes become visible? They are not organized yet, but they're visible in A. So we'll put an A next. And then once that happens and the nuclear membrane breaks down, they tend to then line up in the middle. And so that brings us to C. And then there's a pulling apart step, A, anaphase, that comes next. And then we finish with telophase, B. X are spindle fibers. Y is a nucleus, and you could say that inside there it's not visible, but there's chromatin inside, right? And so chromatin is visible only in D, right? In D, this is chromatin. You can't see it, but that's all DNA. Disorganized. It kind of looks just flat, like there's no distinct dark structures. Where are the sister chromatids available, or where can you see them? I can see chromosomes in A. I can see chromosomes in C. I see chromosomes in E and in B. So chromosomes and sister chromatids are visible in every step of mitosis, from A to C to E, and then finally to B. The only time chromatin is visible is in interphase. That happened actually first. Okay, let's move. Now, just a little quick review. Even though it's the same as the previous one, we're just not characterizing it the same. Let's pull this off to the side so we can see this. Are these cells all the same type of cell? And the answer is currently yes, right? All are plant cells. And they're in the root, right? That's usually where they're found when they're doing active mitosis. Do they have different DNA? No. All cells are identical in their genetic code. Now, where's a parent cell? Well, a parent cell is something like 5 or 6, right? Or comma, 5 or 6, because that's a cell that is not currently dividing, but it could in the future. What does a parent cell divide into? Those are referred to as daughter cells. And the best place to see a daughter cell, kind of visualize it in this picture, is this is the beginning in telophase of the beginning of this will become a daughter, and this half of the cell will eventually become a daughter cell. They haven't finished the dividing yet. And what happens after the cell cycle, after the cell divides, is the cell keeps going around the clock, and we're going to say returns to G1 phase. It goes back to being a plant cell, doing its job, making proteins, bringing in energy, making ATP, all the things that make a living organism a living organism. So remember, just because you're done with cytokinesis and mitosis, you'd then go back and circle around again, and that's back a few slides if you want to review. Okay, why do cell cycles have to exist in the first place? Well, the 3 R's. Okay, what's this guy doing? Well, this person's arm is broken. So one of the reasons why we need to do cell division is to repair broken cells or damaged cells. The second R is this baby who's pretty young, and it is for the purpose of regenerating, right? New cells and an embryo, repair, regenerate, and the last component is to replace older cells. And so the lady who's clearly been overexposed to sun, her complexion is, her eyes were protected by her glasses, but she has some peeling to probably do, and so mitosis will repair and replace, simply replace those cells.